Welcome to Optimizing the Extraction of High Molecular Weight DNA from Tissue, presented by BioCompare and sponsored by Covaris. My name is Tamlin Oliver, Managing Editor of BioCompare, and I'll be the moderator for this webinar. Today, Michelle Kim, lead scientist at Circulomics, will talk about extracting high molecular weight DNA from mammalian tissue, a challenging process that she says can be made easier with the right technology. She'll be joined by Tom O'Hare, U.S. Mid-Atlantic Territory Manager at Covaris, who will provide a brief introduction to Covaris and its technology. They'll get started shortly, but first I want to go over a few housekeeping items. We'll hold a live Q&A session at the end of the presentations. You can submit a question for the Q&A session at any time using the Ask a Question tab in the upper right corner of your screen. The right side of the screen also features an overview of the webinar, as well as more information on the speakers. If you have any technical difficulties at any time, please click on the Test Your Connection button at the bottom of your screen. From there, you can access webinar support. Also, we've provided social media widgets beneath the webinar window to facilitate the sharing of this webinar with colleagues. All right, with that done, I can introduce our first speaker, Tom O'Hare, properly. As the Mid-Atlantic Territory Manager for Covaris, Tom has an active role in supporting scientists within his region for their workflows, including proteomics, cell lysis, chromatin shearing, and NGS applications. Before joining Covaris, Tom was a senior genomics sales specialist at QIAGEN. He received his PhD in genetics from the University of California, Davis, studying telomere biology using the chicken as a model organism. Tom will speak first with a brief overview and then introduce Michelle, who will discuss the use of Nanobind technology to extract DNA from mammalian tissues, as well as show examples of how the DNA performs in long-read sequencing. Welcome to both of you. Tom, please get this webinar started.
Thanks, Tamlin, for the introduction. Welcome, everyone, to the webinar brought to you by BioCompare, sponsored by Covaris. I'm Tom O'Hare, U.S. Mid-Atlantic Territory Manager at Covaris. After a quick intro on Covaris technology, Michelle will present on high molecular weight DNA extraction. Please submit questions and comments throughout the presentation, and we will answer during the Q&A session. Covaris provides tools and technologies to improve pre-analytical sample preparation. Covaris instruments, consumables, and reagents are used in a variety of applications, ranging from tissue processing to DNA shearing. Founded 22 years ago, Covaris developed a technology known as AFA, Adaptive Focused Acoustics, which is used globally in multiple applications. Covaris has several AFA-ultrasonicators in addition to the cryoPREP for sample preparation. Covaris instruments are available for all levels of sample preparation throughput. Do you find sample preparation to be a hassle and an annoyance? There are many challenges with sample prep, and Covaris works with labs on solutions to simplify the process and provide high-quality, consistent results. cryoPREP is one of the Covaris instruments that can provide a solution to sample prep workflow challenges. Michelle's presentation will show the use of the cryoPREP in the Circulomics workflow, producing high molecular weight DNA. cryoPREP is a calibrated benchtop instrument that provides consistent pulverization of frozen tissues in a fast, temperature-controlled process. With the press of a button, the cryoPREP pulverizes frozen samples. Covaris has established protocols for extraction of proteins and high molecular weight DNA from tissue sizes of 10 milligrams to 2 grams. In addition to cryoPREP, Covaris is well known for AFA-ultrasonicators. AFA technology is computer-controlled with tunable acoustics operating outside the human audible range in a temperature-controlled environment. AFA instruments are present worldwide and in thousands of peer-reviewed articles. AFA is used for many applications, including chip, NGS, and proteomics. Today, Dr. Michelle Kim will present on Circulomics Nanobind high molecular weight DNA extraction from tissue using Covaris cryoPREP for sample disruption. Dr. Kim is currently a lead scientist at Circulomics in Baltimore, Maryland. While at Circulomics, Michelle has led the launch of several DNA extraction products. Prior to Circulomics, Michelle completed a PhD and postdoc at Johns Hopkins School of Medicine. The extraction product development that Michelle has led at Circulomics is impactful to long-read sequencing applications, and I look forward to hearing her interesting presentation. Welcome, Michelle, and thank you for presenting today.
Thanks, Tom, and thanks, Tamlin, for the introduction, and thank you to everybody who's here today to learn about extracting high molecular weight DNA from mammalian tissue using Covaris cryoPREP for sample disruption and Circulomics Nanobind for DNA extraction. As Tom brought up, I know sample prep can be not a desirable activity for many, but we hope that our products will make it easier for you. So, Circulomics. We are a biotech company in Baltimore, and we focus on products for long-read sequencing, as Tom mentioned. Our products also help for optical mapping, and we help people trying to use technologies such as Oxford Nanopore, PacBio, and 10x Genomics. We have two main product lines. One is for DNA extraction, for which we presently offer 3 Nanobind Big DNA kits. The first launched kit is the CBB kit, and you can use this to extract DNA from cells, blood, and bacteria. We also have a plant kit, which is for isolating DNA from plant nuclei. We are also currently developing a plant kit that can be used for plant tissue directly without the need to first isolate nuclei. Our third kit is for DNA extraction from tissues. While it was primarily developed for extraction from mammalian tissues, it can be used for extraction from a wide variety of tissues. Our other main product line is a suite of short-read eliminator kits for size selection at different molecular weight cutoffs. In addition to the products we offer, we also work closely with our customers to guide them in all aspects of their DNA extraction, library preparation, and any QC related to sequencing. Today, I will be focusing on DNA extraction from mammalian tissues. I will demonstrate a method we use for disruption, the results one can expect from Nanobind DNA extraction and size selection, and then show you examples of how this DNA performs in long-read sequencing on Oxford Nanopore MinION. The first critical element in extracting high-quality DNA is the tissue going into the extraction. One consideration is the type of tissue it is. For instance, is it fibrous? Is it difficult to break down? Does it have a lot of connective tissue? Does it have high or low nuclei density? Another factor is the quality of the tissue. Was it frozen soon after collection? Has it been stored in any preservatives? Was it exposed to unfavorable conditions before preservation? As a general rule, if the tissue going into the extraction is low quality, the DNA coming out will be low quality as well. Adequate disruption of tissues is another important factor. Some tissues can be quite difficult to disrupt using conventional methods, and unfortunately, tissues that are not well disrupted may require longer lysis periods or result in diminished recoveries and purities. The cryoPREP fully pulverizes frozen tissue samples to a powder and thus alleviates such concerns. A third challenge is the ability to isolate DNA of good purity and size. The nature of DNA isolation with Nanobind allows for thorough washing without DNA shearing. As a result, Nanobind extracted mammalian tissue DNA is typically very high molecular weight with purities that fall within the ideal ranges. Last, I will show you some MinION sequencing results using Nanobind extracted DNA from human tissues that were disrupted using cryoPREP. In addition, you will be able to see how short-read eliminator size selection further enhances read lengths. DNA isolation with Nanobind is a straightforward lyse, bind, wash, elute process. The extraction can all be performed in microcentrifuge tubes and with basic lab equipment. The salient feature is the Nanobind disc, which is a 5mm disc coated with silica. With scanning electron microscopy, we can see that the surface of the disc is covered with microscale wrinkles that themselves are comprised of nanoscale flakes. These flakes contribute to an extremely high surface area, giving Nanobind an extraordinarily large DNA binding capacity. In fact, in many extractions, at the binding step, you can actually see the DNA bound to the disc, which I always find cool, even now. As I mentioned earlier, the nature of DNA binding to nanobind facilitates washing, which you can see is possible due to the loose conformation of the DNA when attached to the disc. While cells, blood, and bacteria can start directly with lysis, the tissues must first be disrupted. For tissue disruption, we use the Covaris cryoPREP, which is a dry pulverizer that uses high-force impact to reduce frozen tissue pieces to powder. We typically aliquot 1 extraction's worth of tissue into 1 tissue tube and use the maximum setting at 10 impacts. Incorporating the cryoPREP into the Nanobind extraction workflow, one can complete a DNA isolation in about 2 hours. More than half of this time is hands-off incubation time. There are several advantages to this workflow. One is the ability to do high-throughput sample processing. Multiple tissue samples could be pre-aliquoted into separate tissue tubes for efficient sequential pulverization. CryoPREP disruption is highly reproducible from impact to impact and user to user, thus eliminating variability from user inconsistency. Since each sample is contained within its own tissue tube, there is a low risk of sample cross-contamination. If DNA isolation will not be carried out right after tissue disruption, the pulverized sample and its tissue tube can be stored in a freezer for extraction at a later time. And last, it is quite easy to operate the cryoPREP, requiring just the push of a button. We used 5 different human tissues to demonstrate the workflow using cryoPREP for disruption and Nanobind for DNA isolation. 5 tissues are human breast, skeletal muscle, colon, kidney, and uterine fibroid. These samples were frozen in a -80 freezer soon after collection and without the addition of any preservative solutions. These tissues vary widely in nuclei density, texture, and ease of disruption. For instance, the breast tissue has a nearly liquid consistency when thawed, while uterine fibroid has a tough, somewhat rubbery texture that is not conducive to disruption. Using cryoPREP, both were easily pulverized to powder using our recommended conditions. For each extraction, we used 25 to 50 milligrams of input tissue. Each resulted in ample DNA for long-read sequencing library preparation. The comparatively low recovery with skeletal muscle, in blue, is due to its low nuclei density relative to the other tissue types. I'd also like to point out the high recovery of DNA from the breast tissue. Breast is illustrative of the importance of carefully selecting your sample. Most of the breast tissue is composed of fat, and if the sample is not carefully chosen, the DNA recovery will actually be quite low due to the low nuclei density of adipose tissue. This breast sample was taken from the ductal portion of the organ, which has a very high cell and nuclei density. Next, we assessed the purities of the DNA and found that they all had ideal UV absorbance ratios, with 260-80 measurements between 1.8 and 2.0, and 260-30 measurements between 2.0 and 2.2. We analyzed the size of the DNA using pulsed field gel electrophoresis and saw DNA from all 5 tissues surpassed 300 kb. We prepared libraries using Oxford Nanopore's ligation sequencing kit and then did 48-hour sequencing runs on Oxford Nanopore MinION. For the 5 tissues, we got an average of 9 gigs of data and an average read length N50 of 27 kb. On 3 of the tissues, we used our short read Eliminator kit to size select and deplete short DNA species from the sample. The short read Eliminator, or SRE as we call it, uses a centrifugation procedure that is similar to standard ethanol precipitation techniques. SRE buffer is mixed with your DNA sample, and the solution is centrifuged to pellet the desired DNA, leaving the smaller species to remain in the supernatant. After a quick wash of the pellet, the DNA is resuspended and ready for downstream applications. The gel image on the right demonstrates the effect of the 3 versions of SRE that we offer, each with increasing molecular weight cutoffs. The utility of treatment with SRE is best illustrated by histograms of the read length distributions from the sequencing runs. Without SRE, the read length N50s were 36.2 kb for breast, 23.1 for skeletal muscle, 28.6 for colon, 18.4 for kidney, and 28.3 for uterine fibroid. We treated the DNA from colon, kidney, and uterine fibroid with SRE and made new libraries and did 1-hour MinION sequencing runs. We saw significant increases in read length N50s for all 3 samples. Treatment with SRE increased N50 for colon by more than 23 kb and for kidney and uterine fibroid by 13 kb. In summary today, I have shown you how using the Covaris cryoPREP for tissue disruption with Nanobind for tissue DNA extraction is a synergistic combination. CryoPREP affords rapid and reproducible tissue disruption. It allows multiple samples to be processed easily and offers a low risk of contamination between samples. Nanobind isolates high molecular weight tissue DNA that surpasses 300 kb and results in DNA with high yield and high purity across diverse tissue types. This DNA can be used for long-read sequencing to generate long read lengths and high throughput, and read lengths can be further enhanced by short-read Eliminator. With that, I'd like to thank you for your time and thank my colleagues at Circulomics, Jeff Burke, Duncan Kilburn, Renee Fedak, and Kelvin Liu, whose work contributed to the development of all of our pilots. I'd also like to thank Hamid Koja, who trained us on how to use the cryoPREP and with whom we continue to work on other projects. We have a collaborator, Wilfredo Mojica, who provided many of our human tissue samples, and I'd like to thank the NIH whose funding has supported our research. And now I will open it up for questions.
Thank you, Tom and Michelle, for those informative presentations. Before we start our Q&A session, we have a few polling questions to gauge where you are with DNA extraction. So bear with me a minute while I get those up. Okay, so the first question is, how are you performing extractions of DNA from fresh tissue? Mortar and pestle, Downs homogenizer, sonication, or something else? So I'm going to give you a little time to answer that, and then we'll share the results. All right, Michelle, can you see the results? It seems like 26% use mortar and pestle. Does that make sense to you?
Yes, we do have a lot of customers who use that, and that's typically a piece of equipment that a lot of labs have. We often use in our own hands a Downs homogenizer or something akin to the tissue vector. We like those because they seem to be more reproducible from user to user, and they don't require liquid nitrogen the way a mortar and pestle do. Mortar and pestle is great, but again, as I mentioned, it can be difficult to get your hands on liquid nitrogen. It can be a little difficult to get you need to practice, I think, from what I understand, to get to a point where you can grind it really well. And you can achieve the same result using a Downs homogenizer or tissue vector. So we do recommend that, actually.
Okay, let's move on to the next poll. All right, the question is, are you satisfied with your method of high molecular weight DNA extraction from tissue? Yes, no, I'm not getting enough long DNA, or I need to know more about Covaris, about the Covaris cryoPREP system. All right, I'll give you a minute to answer that. All right, so it seems that 33% are saying I'm not getting enough DNA, and 55% want to know more about the Covaris cryoPREP system, and then 11% answered no. Michelle, do you have any comments on these results?
Oh, yeah. We talked to a lot of people who have trouble getting high molecular weight DNA. And I think that a large part of it is just, it's not something that a lot of people do. It's a lot of people are getting high molecular weight DNA to do long read sequencing, but it's not like they're doing it every day. So it's very understandable. Anybody is free to reach out to me. We will help you with it. And I think that using the cryoPREP definitely will help because disruption, as I mentioned, is a very critical feature of getting your final DNA of high size And another thing is that a lot of people don't really know what kit to go to, and because there's a lot of differential results that you get from different tissues, that can be very frustrating for users as well.
All right, this poll is, are you working with structural variants such as inversions and translocations? Yes, no, or I will need to do this in the future. So again, I'll give you a couple seconds to answer this. All right, Michelle, it looks like 18% are saying yes, 45% say no, and 36% say they will need to do this in the future.
Oh, that's great. I think that if that is something that you're studying, using the Nanobind will be very helpful because you will have a high likelihood of getting very long, high molecular weight DNA using the Nanobind. And often these structural variants that are being studied require long reads that span more than 100 megs. So, yeah, I think this would be a good approach, and feel free to reach out to me.
Good. All right, we'll go on to the fourth and final polling question before our Q&A. And this one is, are you using long-read sequencing analysis for your analysis? PacBio, Oxford Nanopore, Bionanogenomics. All right, I'm going to give you a second to answer that. All right, it looks like we have 27% using PacBio, 36% using Oxford Nanopore, and 36% using Bionanogenomics. Does that make sense, Michelle? Is that what you're seeing as well, pretty much?
Yes. And even though in my talk I only mentioned sequencing using Oxford Nanopore, our DNA is great for using PacBio or Bionanogenomics. And of course, it's also great for if you're doing DNA applications that require smaller DNA. We work with all of these groups, and we support all our users for whichever end sequencing platform they would like to use.
All right, now we're ready to segue into our Q&A, and Michelle will be joined by Mary Pingatore from Covaris, as Tom couldn't be with us for the Q&A. So, Michelle, we have a bunch of questions for you. So we'll start with the first one. What percentage of the reads that were sequenced were over 100 kb?
So that depends on the tissue type, but generally they were about 5 to 10% of the reads were above 100 kb. And there are ways that we can push the library prep to get more reads at more than 100 kb. We are also currently working on protocols to increase the size of high molecular weight DNA that you're getting from tissues.
I also forgot to mention that you can continue to submit questions, and we'll answer as many as we can today. And then whatever we don't get to, Michelle and Mary can probably get you via email. So please continue to submit your questions. All right, here's the next one. How does the cryoPREP work? Vibration, AFA, what else? And how are the samples, how many samples can be treated in parallel?
So I guess the best analogy is that it's like a hammer. And you put your tissue in a thin plastic bag, and the bag is placed against a solid surface, and then you initiate the impact, and it's metal on metal, and it pulverizes quite quickly.
Okay, the next question is, when are the plant directly extraction kit available?
We are working on that, and I don't have a date, but we hope to be able to launch it quite soon.
Okay, how can I efficiently isolate workable amounts of megaplasmids from bacteria? Is it a low copy number plasmid?
We do have a process for isolating plasmid from bacteria, and for that process, we were using bacteria that had high copy number and mid-copy number plasmids that were about 8 to 12 kb. We haven't tried bacteria that are carrying megaplasmids though, but I can get back to you with some more information on that later.
Thank you. Can we use the Covaris cryoPREP and Nanobind for DNA extraction of Drosophila fly? I'm not even saying that, sorry.
Mm-hmm. So we do have a process for getting DNA from Drosophila. We have customers who have sequenced it and have had good results of that. I haven't used the cryoPREP to disrupt the Drosophila, but feel free to reach out to me. I can share our process for Drosophila extraction.
Okay, here's another cryoPREP question. What size tissue samples are you starting with for your cryoPREP sample workflow?
Generally, we recommend our users always start with approximately 25 milligrams of tissue. And as I mentioned in the talk, different tissues will have different nuclei density. So for some tissues, 25 might be too much. For some tissues, it might not be enough. But we always recommend starting with 25.
Okay, should the tissue samples be pre-cut before processing in the cryoPREP like usual tissue lysers?
It depends on how you got your sample. So sometimes we get samples and they are not frozen yet. And so I will aliquot them into single extraction amounts and give them a good chop with a razor blade or surgical scalpel before freezing them. For some people, they don't want to thaw their sample because they don't want to give it a freeze thaw. So they might just have a solid chunk that can't be chopped first. I think both are fine to go into the cryoPREP with.
Okay, Michelle, we're going to give you a break and throw Mary a question. Hopefully you can answer this one, Mary. If not, I guess Michelle doesn't have a break. But after the cryoPREP, we still need to use PCR, right or wrong?
Well, that's completely up to you and your goals for your downstream workflow. If you need to amplify, then you're certainly free to use PCR. The cryoPREP itself is kind of agnostic. It fits with many different workflows, including PCR.
Okay, Mary, I'll try to throw you another question. And again, feel free to hand this off to Michelle. What is the minimum amount of tissue that can be used with the protocol described?
I was going to say, Sajel already mentioned 25 milligrams, and you can easily go down to 10 milligrams, and we do have larger size cryoPREP tubes that will go up to 2 grams. So the size of the samples can be quite flexible.
Okay, Mary, I guess this one will go for you. Is the cryoPREP effective for bone samples?
You can use the cryoPREP with bone samples, yes, or say tumor samples with calcifications within them. So the cryoPREP works by putting your sample in a small bag and then putting the entire bag into liquid nitrogen to freeze your sample. And that makes the sample more fragile, more easily broken when you put it in the cryoPREP itself.
All right, here's one more question, and then I have a slew of them from Michelle. So, Mary, here's one for you. What about using bead beading for homogenization? How does that impact the integrity and read length?
That's probably a question more so for Michelle, since we don't have or support a bead beater at Covaris. Michelle, would you like to comment on that one?
Oh, sure. It entirely depends on what solution you're using for bead beading. If you're bead beading in a solution that is lysing your cells open, that's probably not going to be good because that's going to end up shearing your DNA. If you're bead beading in a, say, for example, PDS, then that should not lyse open your cells. However, that is a quite harsh treatment because we find that people who are using bead beading will bead bead for quite a long time. So we typically don't recommend that, but obviously it's a fine process. I think you just have to fine tune it for your own sample to get the right bead beading solution and the right duration so that you're not shearing your DNA.
Thanks, Michelle. Here's another one. Does your technology work with FFPE tissue?
We are currently working on that, actually. Yes, we're working on it. The tissue part, the extraction process is the same as our standard tissue extraction. And what we are trying to optimize now is how you get your tissue out of the paraffin, make sure you're getting all the paraffin out. How do you deal with the cross-links? And of course, because it's been dehydrated through the treatment to make the FFPE block, it's going to do stuff to the nucleic acids in there. So we are currently working on a way to get good quality DNA out of FFPE samples.
Yeah, I'd like to say that you can also reach out to Covaris to get more information on extraction of both DNA and RNA and even proteins from FFPE samples by using our AFA ultrasound equipment for emulsifying the paraffin without using organic solvents. So if you're interested, please reach out to us. We'd be glad to talk to you in more detail.
Okay, thank you, Mary. Michelle, back to you. Is there any kit from Circulomics that can handle full-length RNA better?
That is another product we are working on. We don't have one currently, but if you want to reach out to me, we can talk to you about that.
Okay. Is there a detrimental effect to storing samples at minus 80 degrees after cryoPREP?
To my knowledge, there isn't. In fact, we have tissue samples that we have gotten from customers or collaborators that they've themselves had in minus 80 storage for, I think, unknown amounts of time. It could have been a decade. It could have been longer. And while there is some degradation to those tissues that are decade old, in-house on the tissues that we use for R&D, some of them go through a couple of freeze thaws. They've been cryoprepped. They're stored as powder. They're stored in the original tissue form. I don't see an appreciable degradation to the DNA that you extract from that, because the cryoPREP just changes the form of the tissue. So instead of being solid, it's in a powder. But as long as the cells themselves are staying intact and they're always kept cold and frozen, which is what happens when you're using the cryoPREP, I feel that it should be fine to store it for a long time at minus 80.
Okay, thank you. Another question, how does Nanobind work with samples with a high polysaccharide content?
I assume this, well, I don't assume anything, but maybe this person is working with insects or some plants. We have different buffers that are not in the tissue kit that deal with samples that have high polysaccharide content. So that's, we have different additives to the buffers that help shuffle those away from the DNA.
Okay, the questions keep coming in, so I think I have 6 or 7 more for you. So hope your voice holds out. What is the quantity of tissue required with Nanobind and cryoPREP?
So generally, if you're doing one extraction, we recommend 25 mg. And Mary said that the minimum for cryoPREP is, I believe you said 2 milligrams? Or did you say, you said 10 milligrams, sorry. I was just going to say that is a minimum I guess you would want to have in that bag, because you need enough tissue in that bag to actually be in the impact zone. As far as the Nanobind goes, the minimum amount will be guided by the kind of tissue. For example, if you are extracting from, say, spleen has an exceptionally high nuclei density. It's very cell dense. The cells are small. So in fact, you could get, I think, maybe 10 micrograms from a 2 mg input of that tissue. So it's flexible, but we always say start with around 25 milligrams.
Thank you. Mary, I'm going to throw you another question. Obviously, if it's better for Michelle, please just redirect. What tissues are the most difficult to extract from?
Well, we already had a question about extracting from bone, and bone is one of the most difficult to extract from because the tissue is so hard. We do have an ultra thick tissue tube bag that helps with hard samples to keep them from piercing the bag. Another type of tissue that is difficult is skin tissue. Because it is also very hard and kind of a fibrous sort of tissue.
Okay, Mary, maybe you can answer this one too. How viscous is the DNA after extraction, and how do you deal with it if it is too viscous?
I am going to take that one and pass that one to Michelle. Michelle, is that
sure. Yes, yeah. The viscosity of the DNA will depend on the size of the DNA and how much DNA is in there. So if you're extracting from a sample that is going to have tons of DNA, 10, 20, 50, 100 micrograms in your eluate, that's going to be pretty viscous. And we usually we do a couple things. Right after eluting that, I will pipette it with a standard size pipette tip and then let it rest at room temperature overnight. If it's particularly viscous, you could let that rest at room temperature over a weekend, so a few nights. And then before we do QC, I pipette it again. So I don't think if it's really viscous like that, it's pretty high molecular weight, and shearing, you're not have to worry about shearing it with a standard size pipette. So if you want to get it into solution, you're going to have to do that. I know it seems a little scary to people. And people always are very gentle with their DNA when they want it to be very large. But if there's a gel-like texture to it, and it's very viscous. Like, don't be afraid. Just pipette it so you can get an accurate measurement of its concentration.
Okay, thank you. I just want to say we have time probably for 4 or 5 more questions. So, Michelle, is there any development planned for metagenomics, for example, looking at feces samples?
Yeah, I love all these questions. They're just totally our jam. We are currently working on a process for metagenomics. We haven't worked with feces samples yet, but it is on our list of things to figure out how to tackle.
Okay, thank you. Is the sample processing reproducible?
Yes, especially with regard to the tissue disruption with the cryoPREP, it's very reproducible because the system itself is automated, and just the nature of that disruption is just high impact. That is very reproducible. With the tissue DNA extraction, it will, as with any kit, it'll take a little practice, but it is quite reproducible. So with the same tissue type, we generally get the same amount of recovery and quality and size. Usually we find that the variations are probably due to the age of the actual sample or the portion of the tissue from which the sample was taken.
All right.
I will comment that the impact that is used with the cryoPREP is adjustable. So laboratories can write standard operating procedures with a particular impact for a particular tissue type and size, and that does help with reproducibility from user to user.
Thank you. Next question is, do the short read eliminators damage the high molecular weight DNA?
Oh, totally get that concern. It does not. You add our buffer and then flick the tube to mix it, and then it just goes into a centrifuge spin. So it's not going to damage the high molecular weight DNA.
So, Michelle, I'm not sure if the next question is a question or a statement. So after I read it, I guess you can decide. But in my experience, use of the magnet bead gives good yields of DNA. Is there anything that you can add to that?
Oh, for sure. The magnet, we like the magnet. Obviously, our Nanobind is magnetic. It makes isolation easier when you can have something that's magnetic. But one issue that could happen with magnetic beads is if you have very high molecular weight DNA, it can be difficult to elute it off of the beads. Sometimes, for example, if your DNA sample is very viscous, often it will get so convoluted with the beads that it's difficult to get all the beads off, and then you might lose a lot of your highest molecular weight DNA.
Okay, thank you. The next question is, what is the size of DNA with Nanobind?
That will vary from tissue to tissue. Generally, we see DNA spanning about 50 kb up to hundreds of kb. With slightly altered processing, you can get it to like almost a megabase. Again, that will definitely depend on the tissue type, but I would say generally 50 to 300 is what you can expect.
Okay, next question is a protocol question. Is there a special protocol for isolating high molecular weight DNA from mucosal tissue? And we are using the cryoPREP in our department, but when adding buffer to the pulverized mucosal tissue, we see a lot of clumping.
Let's see. I guess what buffer are they adding to the pulverized mucosal tissue? That clumping could be a lot of extracellular matrix, things that would have to be cleared away. We have a buffer in our process that washes away some of this extracellular matrix and things like that are salivary components or other high carbohydrate elements that are not actually part of the cell. So that's what I would recommend. You could wash your sample before you add this buffer, which presumably is a lysis buffer.
All right, 2 more questions, Michelle, and then you can give your voice a rest. Can we use Nanobind for bacterial plasmid extraction? Yes, we don't have an official process launch, but we do have a process for it. Please reach out to me and we can talk to you about it. All right, I lied, actually. More questions are coming in for you. So here's another one. I'm sorry. Okay. Any development of enrichment method of bacterial DNA from host human background for ONT or PacBio sequencers?
Let's see. So I know that people do this computationally after sequencing, but if you're asking whether we have a way to separate bacterial cells from host human cells, we don't have a process for that at the moment.
Okay, thank you. And truly, this is the final question. All right, how much size will be used for extraction of DNA from human adipose tissue?
Adipose tissue is tough because the cells are arguably big, and therefore in any sample, you won't have a lot of cells, you won't have a lot of nuclei, so you won't have a lot of DNA. I have not optimized a protocol for extraction from adipose tissue. I'm sorry to say, but yeah, I mean, reach out to me. We'll talk about it. I'll help you in any way I can. We can discuss options.
Okay, great. Michelle, thanks for answering all those questions, and Mary too. Really appreciate that. That's all the time we have. I just want to thank Tom, Mary, and Michelle for joining us today, and offer a special thank you to Covaris for sponsoring today's event. This webinar will be available on demand in about 24 hours. You can watch it again or share it with colleagues by visiting biocompare.com. Thank you for watching this webinar today, and we look forward to seeing you at our next event.